Act for TB repurposes existing frontline tuberculosis antibiotics to overcome resistance and improve efficacy. Currently, there are about half a dozen different types of drug delivery systems in use today that typically only increase residence time in the serum or increase water solubility. This delivery system is based on formulating inert, safe components with existing frontline TB drugs, altering cellular processes in order to increase drug uptake rate. Antibiotic resistance is an evolutionary guarantee, limiting options to treat bacterial diseases. 5% of tuberculosis cases are resistant to frontline antibiotics, and this number is rising. 5 of these antibiotic complexes have undergone in vitro preclinical testing against Mycobacterium tuberculosis at the National Institutes of Health. The minimum inhibition concentration values for isoniazid against resistant isolates of the bacterium are immeasurable. However, when adding all 3 components to the drug, the resistant isolate becomes susceptible to the drug again, and MIC values decrease substantially. Further trials are needed to prove safety, ensure the drug reaches the target without dissipating, and to determine dosages and tolerability. And now our I-Corps journey. We're merging medicinal chemistry with microbiology to develop delivery systems for existing tuberculosis antibiotics in order to combat antibiotic resistance. We mix the drug solutions in the lab and then send them up to the National Institutes of Health to test against tuberculosis. That's our team in tech, but this is our ever-changing I-Corps journey. On the first day of I-Corps, my hard work didn't make any sense, and there was a lot of progress to be made. So we got out of the building and started off strong on Embassy Row, where we met with a member from the Ministry of Health at the South African Embassy. Here we learned that 80% of the young adults are infected with TB, and reviving manufacturing plants are looking for new products. We have since been in contact with the South African Medical Research Council. Then we visited the Southeastern National Tuberculosis Center, housing top-of-the-line research and treatment for TB. They facilitated 2 full days of tours and meetings, including one with a Nigerian international student undergoing a very toxic, expensive treatment for a very resistant case of TB, giving us a personal experience with the harsh treatment. We learned that improvements need to be made for treating resistant cases found internationally. We thought our technology had a more simple market entry, but we quickly realized entering the pharmaceutical market is always complex. But in one of our last interviews, we learned that the time to enter the market is now because of its predicted growth in the next 5 to 7 years due to advocacy groups and rising antibiotic resistance scares. We think this technology is a go in terms of seeking more funding to facilitate further in vivo preclinical trials to make a compelling argument to R&D pharma companies and R&D donor-funded programs to license this technology.